What type of scientist am I? I guess the easiest answer is say I want to be thought of as an evolutionary biologist. That's still a generic general term. Um, by the end of this program, I'll be a comparative physiologist. Um, but as of now, I'll definitely say that my the area topic that I enjoy most is evolutionary biology. So I guess that's what I would say right now. I love the comparative aspects of biology most. Um, because there's so many awesome questions you could ask, and like even when you answer one question, it opens up tons of more. Because like cause biology is not it's not simple. It's like it's complex. Like living things don't always move in a predictable manner. So there's a lot of exceptions to the rules, and figuring out why does this exception exist. Are there is this really an exception, or is this more common than we think it is? So I love like reading papers about that. So definitely say those topics is what gets me going in general. Oh, I love like anything that's comparative, whether it be comparative anatomy, comparative physiology, comparing their genetics. I love parsing out like sister lineages and what like what differences are there in the genetic sequences? Are there morphological variations? Are there behavioral variations, ecological variations? So I love looking at like a system and parsing out the differences between them. We work on pricklebacks in our first year, so it's a I can pull up a picture of it. It's like a thin intertidal fish. You could find them like they like go in like crevices and holes in the intertidal. Like as a first year project, Donovan usually has his work on like digestive assays of like previously dissected gut contents and like GI tracts. So it's definitely interesting to compare the differences in diets between them, the differences of their enzyme expression. That and does that correlate with their diets, and how's that bear out and how where we find them, and how they interact with their environment, how they acquire nutrients from their environment. My master's thesis was comparing three different closely related chitin species to each other and figuring out, try to parse out where do they live? Do they live all in the same area? Do they have different geographic ranges? And then looking at their morphology, because people say that it's hard to identify them from each other. So part of my master's thesis was to look at their morphology and figure out, is that true? Are they identical to each other, or do they look different? Once you're able to perform like a molecular assay on them to identify them, then I was able to parse out their morphological differences, or attempt to parse out their morphological differences. So like those type of questions and those type of research areas definitely I enjoy the most. Yes. These are two graphs from my master's thesis. So one of the parts of my thesis was, are there morphological differences that are easily observable between the three species? So I'll go to this graph here on my right first. I was looking at the relationship between the number of gills on their left side and their overall body length. With chitons, it's a general like linear relationship between body length and the number of gills. So I wanted to make sure that they fought like these kinds follow that same trajectory, and like is there a difference between the number of gills between them, or do they have the same number of gills? And I found that there is a difference in the number of gills between the three species. Specifically, Nullina californica, which is the red dots, is a really long chitin, and it has more gills than Fluxa and species A. But regardless of the size range, species A always had fewer gills than either Nullina californica and Nullina fluxa, which is interesting because that's a further question: is why? I didn't investigate why that is, but that's something to follow up. Why did this species of chitin have fewer gills than its sister taxa? So there's a lot of good like conservation implications for my research, because it's hard to develop a conservation plan if you don't really know what species are there, because currently they're all synonymized as one taxa. So if you implement a conservation plan for Netherlina, and you don't know that there's three species instead of one, your conservation plan won't be as effective. Like your conservation plan will like it'll be hard to create a plan first of all because these are three different species with three different with different habitats and different ecology. So it's hard to make a cohesive plan for that first of all. And even if you were able to create a plan, there's no guarantee it will work on all three. It might work on two of them, might just work on one, might not work on any of them. So it's definitely like if there's like a group of cryptic species, this type of work is really beneficial for those who are working in conservation to know like, all right, yes, these species kind of look the same, but we need to know that they're not the same. Like we can't treat them all as one organism and create one plan for everyone. So I think that's probably the best application for this type of research.